When sperm enters the female reproductive system, it travels up to the fallopian tubes, searching for an egg to fertilize. This marks the beginning of potential conception. The sperm penetrates the outer layer of the egg. Only one sperm can successfully break through this protective barrier, and once it does, the egg surface undergoes an immediate transformation to prevent any other sperm from entering. As the sperm enters the egg, its tail is no longer needed and begins to disintegrate. The head of the sperm fuses with the egg, and then the sperm's nucleus, carrying the father's DNA, merges with the egg's nucleus, which contains the mother's DNA. The genetic material from both mother and father combines to create a complete set of 46 chromosomes, with half inherited from each parent. This single cell, known as a zygote, represents the first cell of the developing embryo. It contains all the genetic instructions necessary to form a new individual. The zygote now begins a rapid process of cell division. It divides into 2 cells, then 4, then 8, and continues, forming a small, growing ball of cells. As the cells continue dividing, gentle muscle contractions in the tube's walls, along with tiny hair-like structures called cilia, propel the zygote along the fallopian tube toward the uterus. This remarkable journey typically takes about 3 to 5 days. By the time the zygote reaches approximately 16 to 32 cells, it becomes what's called a morula, which resembles a tiny cluster of cells. By the 4th day of development, the morula begins to absorb fluid, and a large cavity called the blastocyst, or blastocele, forms within it. When the blastocyst finally reaches the uterus by day 5, it hatches out of its outer membrane. The blastocyst has an outer layer of cells that will eventually form the placenta, and an inner group of cells that will develop into the baby. The blastocyst attaches itself to the uterine lining, known as the endometrium. This critical process is called implantation. This allows the blastocyst to secure itself to the uterine wall, enabling it to obtain nutrients and oxygen from the mother's blood supply, supporting its continued growth and development. Now, in rare cases, something quite unusual can occur during the early stages of cell division, the formation of identical twins. The developing cell splits into 2 distinct groups, each with a complete set of DNA from the original fertilized egg. These 2 groups of cells continue to divide independently, and each one will eventually develop into a baby. The exact mechanism that triggers the splitting of a zygote is not yet fully understood. Potential genetic factors, along with certain environmental influences, such as specific fertility treatments, may affect the likelihood of this occurrence. The occurrence of identical twins is often described as an event of random biological chance.